Hello, my name is Aaron Manusov. My partner is Vince Diego, John Blangero, and Sarah Williams Blangero, and we are from the Department of Human Genetics. And what we're going to talk about today is genotype by environment interaction of the Frailty Index. We have nothing to disclose. And our major goal is to assess the genetic components of the Frailty Index. We're going to define Frailty Index and frailty, calculate the Frailty Index based on variables collected in a large Mexican-American study, and we're going to determine if there are gene by environment interactions that may affect the Frailty Index. So what is frailty and what is a Frailty Index? So the phenotype is descriptive and measures what the individual looks like. That's frailty. The Frailty Index describes the variables and contributions to the development of the phenotype. So if you look on the left, there's two gentlemen. One is looks frail, is using a walker, and the other one, same age, doesn't look as frail. And it looks like maybe his VO2 max is better, his strength is better. So that's the phenotype. Frailty Index has a living system that is affected by multiple variables. So if you look on the right, Frailty Index looks at physical health, unintentional weight loss, problems with memory, feeling down, living alone, and it is the sum of deficits divided by the deficits measured. One of our questions is, do different individuals respond differently to the same environment? So why study frailty? The Frailty Index is the most accurate measure of frailty, as it is a systems approach. The Frailty Index predicts cognitive impairment, hospitalizations, morbidity, and mortality. It is a more dynamic measure of frailty over time. Frailty is a multi-system decline and increased vulnerability to external stressors. Frailty places a large burden on individuals, families, and healthcare systems, and frailty can be prevented. So if you look at a basic quantitative model, you have your genes and you have your environment and lifestyle, and that's what makes you. And that's an additive. So your genes and your environment make you. What we're talking about is the environment and the genotype interacting and creating a phenotype. So the environment affecting the genotype. And so the genes don't change, but here's a good example recently published by our group looking at sedentary behavior. Used to be thought that sedentary behavior made you fat and you had a heart attack or a stroke. And in our research, we looked at the environment actually changing the expression of the genotype. So some of the genes are turned on and off, on or off, and the genotype expression changes, and increased inflammatory markers are associated with decreased sedentary behavior. So that's pretty exciting, that it's not just that you are sedentary, it's actually that sedentary behavior turns on and off gene expression, which means that we can do a lot of things to change if we look at an environment interacting with the genotype. First thing we want to look at is our data. And this is a histogram of observed values of the Frailty Index and overlaid with the probability density function, that red line. And what does our data look like? So the histogram is the observed frailty values, and it looks like a pretty normal distribution. And if you calculate, and that's expected, if you calculated a theoretical, that's of the common distribution used to model the Frailty Index, the Weibull distribution was found to have this best fit, shown here as that little red line. And although it may be the best fit, but it also could be fitted to a normal distribution. That's important for our calculations. What is gene by environmental interaction? As I said, genes are not changed by the environment. Gene expression can be altered by the environment. Genes can be turned on or turned off by the environment. And there are many mechanisms by which this can happen. We use a novel statistical model to assess evidence for gene by environment interaction. And it's novel, but it's not new. It was created and designed by one of our senior authors, Dr. John Langero, who developed this a very powerful statistical method, and that's been used by multiple researchers. So let's talk a little bit about gene bioenvironmental concepts so that we can understand as we talk about our findings. So in type 1, you see the genes 1 and 2, the solid line and the dotted line. As you go from one environment to the second environment, they both go the same. There is, they both go 1 and go up, and the second one goes up, and it's the same. You look at type 2, the gene 1 changes as you go from environment 1 to environment 2, but gene 2 doesn't change. Look at 3A. That is a direction. So one is going down and one is going up. And on the opposite, it's divergent. Then you can look at this one. And this shows you that as you go from environment 1 to 2 to 3, that there's a time in environment 2 that they're both the same. So that's important to know when we calculate our gene by environment interaction. So the first thing we need to do is what we're measuring heritable. So heritable is defined as a trait describes how much of the variation in a given trait can be attributed to genetic factors. So you have Jennifer and Karen, they both were identical twins born with genes that absorb fats twice as fast as the average person. Jennifer eats healthy, low-fat food, and her obesity genes are not triggered. Karen eats fatty, unhealthy food, and her obesity genes are triggered, and she is larger. So we hypothesize that there is a gene by environment interaction and is actually essential to the ontology of frailty. And we can use our statistical approach to identify. So if you look on the right, there's multiple different ways that the environment and the genes can interact. You could have methylation, you can have histone modification, you can have toxins, pathogens, drugs, nutrition, and in the environment, and what our team is very interested in looking down here in the valley, and you can have mutations. We calculated the frailty index for a pedigree-based sample of over 1,000 Mexican Americans in the San Antonio Family Heart Study. And we look at 30 different variables, 35 different variables. And we calculated the heritability of the frailty index in this samples. Is there a gene by environment interaction? So in our population, the heritability of the frailty index is 43%. This means that 43% of the variation in the frailty index is attributable to genetic factors. So after detecting evidence that genetic factors influence frailty index, we then search for potential environmental factors that are involved in gene by environment interactions on frailty index. And we found 5 such interactions: total fat intake, total fiber intake, total cholesterol, total protein, and level of education. Look at those p-values of fat and fiber. So what does this mean? The frailty index is a model that looks at frailty as a ratio of deficits. This is the first assessment of gene by environment interaction as it pertains to the frailty index. We use a large data set of variables in Mexican Americans to calculate a frailty index. 43% of variance in our calculation of frailty index is attributable to genetic factors. We have evidence of significant genotype by environment interaction on the frailty index. For certain people, the dietary environment significantly affects the frailty index. We can now evaluate frailty with genome-wide association, transcription, and methylation studies to determine how genetic expression is influenced by the environment. Our studies of the frailty trait will help us target interventions to those most susceptible to frailty. Thank you very much, and I'll entertain any questions.